Hi all. When I think about the muscular system, I think about some of the patients I've had over the years that have had troubles with their muscular system. For example, sometimes a post-op cardiac surgery patient who was cool during their surgery will then have the response of shivering when they're back in the ICU. Shivering is using a lot of skeletal muscle and there's a great deal of contraction going on. So as you can imagine, that changes their metabolism. It increases their metabolism. It increases their oxygen consumption, their CO2 production, and it increases their temperature as well. It's an effort for the body to increase temperature. So from that example, we know then that muscles have a role in warming the body. So that is one example. Another example of patients I've dealt with that have had muscle abnormalities would be quadriplegics who have no tone of some of their muscles. So we know that they would have flaccidity or be flaccid. That means that the muscles are very, very relaxed. All right, so let's get started on this section. We know that we've been using our muscles all of our life for running around and skipping and jumping, and those muscles that we traditionally think of would be our skeletal muscles. You know, when we do a bicep curl, for example, we're using skeletal muscles. But there are two other types of cardiac muscle that are very important, of course. Cardiac muscle, well, we need that in order for our heart to beat, so to pump blood around our body. And smooth muscle, this is a very interesting group of muscles because these smooth muscles are wrapped around hollow organs. For example, think about our blood vessels. Well, our blood vessels always have some tone. They have a little bit of muscle contraction going on. And what about in our gastrointestinal system? How do we move food through? Well, we use smooth muscle. This slide includes a little bit of terminology, such as the term myo, meaning muscle, and sarco, meaning flesh. A lot of the terms in this lecture start with the prefixes myo and sarco. And I think we all understand that the way that muscles function is by contraction or shortening. Fantastic slide that compares the three types of muscles. We've talked about skeletal muscles, like in this example, this person is doing a bicep curl. These types of muscles, they're very long, they have a cylinder shape, they have many nuclei, and they have lines in them that are visible. So it's called striated. Okay, cardiac muscle. So this is also known as myocardium. It is the heart muscle. These are a little bit different. They have a branching shape. We can see that in the image that they have branchings. They have one nuclei, they have striations, which means it has visible lines, and they have a few other little qualities such as intercalated discs, which we'll learn about soon. Smooth muscle, which is around our gut and around our blood vessels, these are a little different. They are fusiform shaped. Some authors will call that spindle shaped. We see that they're wide in the middle and they taper off on each end. They have one nucleus and they don't have those visible lines or striations. You know, I should also talk about another clinical example here. It's a great thing that there are three types of muscle groups in the body. Sometimes if you go to the operating room and a surgeon wants to cut you open in order to perform surgery, the surgeon is going to want your skeletal muscles to be completely relaxed. The anesthesiologist will administer a drug such as rocuronium or a similar type of drug. These are neuromuscular blockers that cause all your skeletal muscles to relax. The really great thing is that those drugs only impact the skeletal muscles. They don't cause, cause paralysis of the skeletal muscles, but thankfully they don't cause paralysis of the cardiac muscle or the smooth muscle. This is a good thing because we want your heart to continue to beat and we want some tone around your blood vessels, for example. Okay, so that's just an aside. Now let's chat a little bit about this image. Skeletal muscle is voluntary. That means that we can control it. It can be slow or fast contraction. And cardiac muscle is involuntary. We don't have to think about that, thank goodness. The heart has its own pacemaker which controls the speed of the heart rate. It's affected by things like the nervous system and hormones as well, but it's the pacemaker that sets the pace. The contractions are slow and rhythmic because they happen over and over and over again, thankfully. Our smooth muscles, again, these are around our blood vessels and around our gut, for example. We don't have to think about these either, thank goodness. So they're involuntary, controlled by the nervous system and hormones, for example. They tend to be slow, rhythmic contractions. Just picture the food moving through your intestines, and imagine how this needs to be a slow and rhythmic contraction to sort of move this along. It's called peristalsis in our GI tract. Let's talk about the tendons that attach skeletal muscles to bones. Well, if you enjoy eating wings, you've probably noticed all kinds of tendons, for example, in the wings. So next time you eat wings, take a look at that connective tissue which connects the muscle to the bone, and you'll know that that's tendons. The term striated and voluntary are mentioned here as well. And very interesting, you know that our skeletal muscle, when we're sitting in our chairs, it always has a little tone, doesn't it? If our skeletal muscles were completely relaxed, we'd just fall out of our chairs, which wouldn't be pleasant. So that helps us understand that skeletal muscles always have a little tone, or what I mean by that is some of the muscles in your skeletal muscles are taking turns contracting a little bit just so that we get to be held upright in our chair. Standing requires even more skeletal muscle use than sitting, and sitting requires more skeletal muscle use than laying down. Also, there are numerous muscles around each of the joints to try to stabilize the joints. And we know already that muscle contraction generates heat, because if we need to warm ourselves up, we can shiver. So the design of the muscle is kind of interesting. It would be like, we know that ropes have many strands in them. So if we took a few ropes and line them up together and then put some connective tissue bands around these ropes, we would be mimicking how the muscle is designed or developed. So what we have are sort of muscle tissues surrounded by connective tissues and all grouped together. So here are some of the connective tissues that are listed. I want to take a moment to pronounce them so you know how to pronounce these. So endomysium is around a single muscle fiber or cell. Muscle fiber and muscle cell mean the same thing. So they are at the smaller level. Perimysium is around a bundle of muscle fibers. Epimysium is around a grouping of those, and then the fascia is on the very outside of the epimysium. So this is an awesome image because we see the relationship between the bone and the skeletal muscle, and we see that they're attached by tendon, which is a connective tissue. There are other connective tissues as well, though. If we look inside of a muscle, the most innermost connective tissue is the endomysium. It surrounds the actual muscle fibers or cells. And then what happens is those muscle fibers are kind of lined up and bundled together into a fascicle, and then that's wrapped with perimysium. And then a whole bunch of those fascicles are bundled together again, and then that is wrapped with epimysium. So that's around the entire muscle. So that's kind of like the relationship that we see with this connective tissue. So here's the muscle that surrounds our stomach, for example, our small intestine, our arteries and veins, and all kinds of our internal organs. If we're to look at this under a microscope, there would be no visible lines, so there are no striations. It's involuntary, meaning we don't have to think about it. Thank goodness, that would take up a lot of brain space. It contracts in a pattern known as peristalsis and segmentation to cause movement within, and they're considered spindle-shaped, and the contractions are slow and sustained and rhythmic. The structure of the muscles around tubes, so smooth muscle, is often in two layers going in both directions. Cardiac muscle is your myocardium, or your heart muscle. Has a very important job, of course, of pumping blood all around our body. It has striations, so it has visible lines, and it is involuntary, meaning we don't have to think about it. And it is special because it has branchings, like the branches of a tree. And it has intercalated discs where the muscles touch each other. And it contracts in, indeed, a steady rate set by the pacemaker, specifically known as the sinoatrial node, or the SA node. This shows the structure of the cardiac muscle. The cardiac muscle is anchored onto the fibrous skeleton of the heart, which makes it sound like it has a bony skeleton. It actually doesn't. The fibrous skeleton is just some strong connective tissue that gives strength to the heart and allows the muscles to anchor somewhere. The muscle cells have a special cell membrane called sarcolemma. The reason why we're chatting about this is because we're going to learn about how the nervous system has a nerve that comes down to and almost touching the sarcolemma, or the cell membrane of the muscle cell. Also, there are myofibrils, which are termed long organelles inside the muscle cell, so we're going to be learning about those. And they create these areas of lightness and darkness that causes the lined look, or the banded or striated appearance of a muscle. So it's because of these myofibrils and the way that they're structured that the muscle looks striated. Now this can get a little bit confusing because we're looking at the structure of a muscle and how it has many smaller and smaller and smaller components. So just to orientate ourselves here, what we're looking at in this image is one muscle cell, also known as a muscle fiber. Okay, so this is an individual muscle cell known as a muscle fiber. It has a cell membrane, which we now know is called a sarcolemma. We see that as the outer wrapping here in this image. Also, there are nuclei inside the cell. Now, looking smaller again, so we're kind of getting smaller and smaller here, we're looking at what makes up a muscle cell, and we see there are more cylinders. Quite interesting. That's just cylinder after cylinder. When we get down to the smallest level, this is made up of myofibrils, which of course we're going to be learning more about. And the way that they're lined up, we've already said, it creates this appearance of darkness and lightness, or these bands. Remember, that looks striated. Oh my, we're getting smaller and smaller with our myofibrils and our myofilaments. So we're looking at the smallest level that we're going to be looking at in the muscle. And this is a myofibril. There would be hundreds to thousands of myofibrils in a muscle cell, just to kind of put it in perspective. So let's look at this myofibril. Well, let's look at what it's composed of. It's made up of myofilaments, thick myofilaments and thin myofilaments. The thick myofilaments are also known as myosin. They have two names. And the thin myofilament is known as actin. If we look at the thick myofilament, the entire length of it is called the A band. It looks dark on appearance. The middle of the A band has an H zone where there are no thin filaments, and it also has an M band, which is the center line of the A band. Now, let's look at the I band. The I band is made up of thin filaments only, and it has a middle line called the Z disc. The sarcomere is a small component of this structure, and it's made up of thin and thick filaments. And this is the actual sort of working unit of the muscle, I would say. It's the contractile unit of the muscle. What is going to happen is during contraction, the thick and thin filaments are going to slide over each other, therefore shortening the length of the sarcomere. And by all of these sarcomeres shortening, we're in turn shortening the muscle. So we're using myofilaments known as thick filaments, they are known as myosin, thin filaments known as actin, and they are overlying one another. Like your fingers, if you part out your fingers and organize them between one another, we could shorten them by making them slide past one another. We know that the thick filament is also known as myosin. The myosin has heads jutting out from it, and they're called myosin heads. When the myosin heads connect to a thin filament, it's called a cross bridge. Also, energy is used in order to activate these myosin heads, which will lead to muscle contraction. We know that thin filaments are also known as actin. Actin are anchored together at a Z disc, they're the midline of the actin filaments. And also just want to mention that when contraction occurs, there's a sliding over of actin and myosin, and the H zone is going to disappear. So I also want to mention that actin has sites on it called myosin binding sites. And at these sites, the myosin heads can actually connect and pull like the oar of a boat, and causes a sliding motion. This is called the sliding filament theory. It's the idea that the myosin heads can join to the myosin binding sites on the actin and sort of row or pull, causing a sliding of these filaments across each other and a shortening of the sarcomere, therefore a shortening of the entire muscle. Also a note on this slide is that there is a mesh network within these cells called sarcoplasmic reticulum. It's just a special type of endoplasmic reticulum, but its really special job is to store and release calcium. So with each contraction of a muscle cell, there's going to be this release of calcium and then a sopping up or a mopping up of the calcium and store it back into the endoplasmic reticulum. Please be prepared to be able to label this on an exam and know these very well because there are lots of test questions on this. Another great image of a sarcomere. Let's take a few minutes to look at this. We see these pink images here in the middle. So those are the myosin. They have myosin heads jutting out from them, and the middle line is called the M line. Then we see the actin in blue. Actins are connected by a Z disc. That would be the middle of two actins. And we see that the length of the sarcomere is from Z disc to Z disc. During contraction, the sarcomere is going to shorten. When all of these sarcomeres shorten, of course, it causes the muscle to shorten as well. There needs to be a mechanism in place to tell skeletal muscles when to contract. So what there is a neuron, a nerve cell, that comes down and reaches out to numerous skeletal muscle cells and tells them when to contract. This grouping of the neuron and the muscle cells impacted is called a motor unit. It's a great picture. What we see here are a couple of motor units. Let's look at motor unit 2. It's in purple. We've got a neuron in purple that's coming along. It's a nerve cell or neuron. It's coming along and it's basically reaching out to numerous muscle cells and it's able to tell them when to contract. There's some terminology here, muscle fibers, those are the muscle cells, and axon terminals at neuromuscular junctions. Okay, an axon terminal is just the end of the neuron, and the neuromuscular junction is the area that includes the end of the neuron and the sarcolemma of the muscle cell. Of course, here we have some of those definitions. So the neuromuscular junction, big fancy word, but it just means the terminal end of the motor neuron, and it's meeting the sarcolemma of a muscle. And this is where information is going to be exchanged in order to tell the skeletal muscle to contract. There's a synaptic cleft, which is a small gap between the end of the neuron and the sarcolemma of the muscle. So these things don't actually physically touch each other. The neuron and the muscle cell don't touch each other. There's a little tiny gap in between. And in that gap, a neurotransmitter can be released. The neurotransmitter is like a chemical messenger. In this case, it's acetylcholine that is used. So what happens when this nerve impulse moves down the neuron and to the axon terminal, the end of the neuron? Well, calcium channels are going to open and calcium ions are going to enter the axon terminal, which is the end of the neuron. The calcium ion entry causes synaptic vesicles, which are little sacs inside the end of the neuron, to release acetylcholine. We've learned that acetylcholine is a chemical messenger. It sends a message across the synaptic cleft. So step 3, the acetylcholine diffuses across the synaptic cleft and attaches to receptors on the sarcolemma of the muscle cell. So that's just the wall of the muscle cell, the cell wall. If an adequate amount of acetylcholine goes across the synaptic cleft and joins with the sarcolemma, it's going to cause the sarcolemma, the muscle cell, to be more permeable to sodium ions. Also, potassium ions will diffuse out of the cell, but more sodium ions will enter the cell than potassium ions leaving the cell. This causes depolarization. It causes a change in charge of the cell membrane, thereby opening even more sodium channels. When even more sodium channels open, we have further depolarization, and then it's termed action potential. Once the action potential occurs, it's unstoppable. The message is definitely going to happen across the muscle cell. This electrical impulse is conducted from one end of the cell to the other. The next step is that the acetylcholine needs to get broken down. It's broken down by acetylcholinesterase. and this ends muscle contraction. So how then does the muscle cell reset and get ready for a new contraction? Well, this is how. It returns to its resting state because potassium ions will continue to diffuse out of the cell, and a sodium-potassium pump will use ATP to pump sodium and potassium back to their original positions. I recommend printing off a full-size page of this slide. It hits on many of the previous slides in the discussion that we've just had. It talks about, at the very top, how there is a nerve, and the end of that nerve almost touches the muscle cell. There's a synaptic cleft in the middle. Acetylcholine is released there as a chemical messenger, and acetylcholine esterase is the thing that breaks down acetylcholine. And then it goes on to talk about when the nerve impulse reaches the axon terminal of the motor neuron, in point number 1, that point number 2, calcium channels are going to open and calcium is going to spill into the axon terminal. Number 3, this calcium entry causes the synaptic vesicles to move out to the edge and release their contents, which is acetylcholine. And number 4, the acetylcholine diffuses across the synaptic cleft. Remember, it's a messenger. And number 5, it binds to the sarcolemma, and it causes an exchange of sodium and potassium, which causes depolarization. And then we have the acetylcholinesterase breaking down, the acetylcholine. Once enough acetylcholine passes across the synaptic cleft, an action potential will be reached. This image gives us the idea that it spreads very rapidly across the muscle cell and affects the entire muscle cell. It's called an all or nothing phenomenon.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
This video is excellent. I would recommend playing it a few times, making yourself your own notes following along. I think that that would help you with your comprehension of this physiology, which is a little bit complicated. So we're going to talk about the sliding filament theory. We'll be talking about myosin and actin. They overlay one another, and the idea here is we want them to slide across each other to shorten the muscle. So how does that happen? Well, for one thing, myosin needs ATP. Once myosin gets ATP, the myosin head gets cocked into position like setting a springy rat trap, I guess. So now this is cocked into position. Now, what about the actin? Well, actin has something on it called myosin binding sites, which are just like what they sound like. They're a spot for myosin to connect, the myosin heads especially. The thing about these myosin binding sites is that they are eager to connect with the myosin head, but they have bodyguards known as troponin tropomyosin complex, which cover them. So what happens here is calcium is going to get released from the sarcoplasmic reticulum and bind to the troponin on the actin filament. This causes the troponin tropomyosin complex to move out of the way of the myosin binding sites. Now these sites are available and ready for the myosin heads to connect. So the myosin binds to the actin and it pivots, just like when you have that pulling motion of rowing in a boat. It causes the thin filament to slide towards the center of the sarcomere. This is the sliding filament theory in action. The contraction occurs and the cell shortens. When all of these cells shorten, it causes the muscle to shorten as well. During a contraction, a cross bridge attaches and detaches several times. Okay, the term cross bridge is when the myosin head is connected to the actin. Just the last note, ATP provides the energy for the sliding process, and it will happen as long as calcium ions are present. At the end of contraction, it's time for the calcium ions to be put away. They are going to be mopped up by the sarcoplasmic reticulum and stored for the next contraction. Here's the sarcomere relaxed on the top and contracted on the bottom. Notice that the H band disappears when the sarcomere is fully contracted.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
This is another fantastic video. It's all animated. It's so well done. Again, I'd recommend listening to this a few times, making your own notes in your own language so that you understand this process. Each muscle cell has an all or nothing principle in terms of contraction. So once it's stimulated enough to contract, the entire muscle cell will contract. However, when we look at the entire muscle together, we could have a graded response, which means that the skeletal muscle can shorten at different levels. For example, I may contract my bicep to move my hand just an inch, or I may contract my bicep to make my hand actually touch my shoulder. So that would be different degrees of skeletal muscle shortening. It's a graded response. Know that the muscle needs ATP as its energy source in order to energize the myosin head. So what happens is there's only a little bit of ATP stored in the muscle fibers, and it gets used up really quickly. So there are three pathways to regenerate the needed ATP. There's direct phosphorylation, the aerobic pathway, and the anaerobic pathway. So we're going to take a look at each of those. The first one is pretty straightforward. I think of it as exchanging Canadian and American money. $1 Canadian is exchanged for approximately $1 American. It's different currency. So it's called direct phosphorylation, and what happens is inside of our muscle cells we have a little storage of creatine phosphate, and it stores energy. And after some ATP has been depleted, ADP remains, adenosine diphosphate. It has two phosphates on it. The creatine phosphate can give up one of its phosphate groups to the ADP to regenerate it back to adenosine triphosphate again. So we're giving it a phosphate in order to make it three phosphates again, adenosine triphosphate. The creatine phosphate supplies are exhausted in less than 15 seconds, so that little stash of energy is used up pretty fast. And for each creatine phosphate, we get one ATP produced. Oh my goodness, this is such an important point. This slide has very, very important information. Many of the patients I've dealt with over the years have had episodes of not enough oxygen delivered to their tissues. During those episodes, their body had to use anaerobic metabolism in order to supply ATP for itself. We will learn in this slide that anaerobic metabolism produces lactic acid. Lactic acid is an acid, so it lowers pH. I have seen many, many times patients with an accumulation of lactic acid in their body because of anaerobic metabolism. This accumulation of lactic acid has led to a low pH. The low pH is called acidosis, low pH in their blood. This is detrimental to the patient, and it can actually be life-threatening. So let's see, why is that lactic acid produced? Okay, so anaerobic metabolism. Ana means no, and aerobic refers to oxygen. So no oxygen, we're going to have anaerobic glycolysis and the formation of lactic acid. This reaction, it breaks glucose down without the presence of oxygen. Glucose is broken down into pyruvic acid, and 2 measly ATP. That's all we're getting now from the glucose. The pyruvic acid is converted to lactic acid. Lactic acid, yes, it causes muscle soreness, but beyond that, too much lactic acid is going to make the patient have an acidosis, which can be dangerous. This reaction is not very efficient. We're only getting 2 ATP for each glucose, but it is super fast. And obviously we need a ton of glucose then in order to get the ATP we need, because it's so inefficient. This would be used if you were an athlete and you were sprinting, for example. During a sprinting activity, not enough oxygen can be delivered to the tissues, so anaerobic metabolism needs to occur. And it is one healthy way of training. But in our patients, it becomes an unhealthy process. There's too much anaerobic metabolism sometimes. And like I say, it leads to an accumulation of lactic acid and a low pH in our patients, or acidosis. Just a fantastic image again. I like these diagrams showing that we are going to do an anaerobic pathway, which means without oxygen. And what happens is the glucose is getting broken down by glycolysis. This is occurring in the cytosol. Very important to note that. So it's not inside the mitochondria, it's in the cytosol of the cell. 2 ATP are produced and pyruvic acid. Pyruvic acid is going to turn into lactic acid, and this lactic acid is what causes an acidosis potentially. This is the pathway that occurs if no oxygen is present. Now in the next couple slides we're going to learn what happens when oxygen is present. In that case, the pyruvic acid takes a different path and actually is going to produce even more ATP. Here is an excellent way for us to produce ATP for muscle contraction. It's called aerobic respiration. Aerobic means with oxygen, so we have to have oxygen present in order for this to occur. It supplies most of our ATP at rest and during light exercise or moderate exercise. It uses a bunch of pathways, and together they're called oxidative phosphorylation. Of course, oxygen has to be present, and this is going to occur inside the mitochondria. Remember, the mitochondria is the powerhouse of the cell, where most ATP that is used is produced. So what happens? Well, glucose gets broken down into carbon dioxide and water, and 32 ATP are produced. I'm going to say approximately 32 ATP because different textbooks differ slightly on that. Also, remember that oxygen needs to be present. This is generally a slower reaction. I really like this image because it adds a little tiny bit more detail about this process. Note that the glucose first got broken down to pyruvic acid. That process was actually called anaerobic metabolism, and it yielded 2 ATP. Now the pyruvic acid needs oxygen to be present, fatty acids and amino acids, and this all needs to happen inside of the mitochondria. This process, oxidative phosphorylation, can now occur inside the mitochondria. It's termed aerobic respiration. We're going to have the production of CO2, water, and 32 ATP approximately. Sometimes I have a chance to walk up a very steep hill from the hospital to UNB St. John campus. Well, it's not very steep, but it feels very steep when I get to the top. It's about 4 stories. When I walk up that hill, by the time I get to the top, I'm breathing deeply and rapidly. And this slide is going to talk about why that happens. So if we have muscle activity that's strenuous and prolonged, we'll say that this walk that I do is about 4 or 5 flights of stairs, and I usually do it very quickly. What is thought to happen is that there becomes an ion imbalance in the cells of calcium and potassium. There's an oxygen deficit. We have too little oxygen in the area, an accumulation of lactic acid from the anaerobic metabolism, and not enough ATP is able to be supplied to the muscle tissues. This makes us feel pretty fatigued. But also, once we get to the top of that climb, we have an oxygen deficit. It means that we owe oxygen back to the area, and it needs to be repaid by breathing rapidly and deeply to ensure that we have sufficient oxygen into our bloodstream. I think it's pretty great that we always have some muscle tone going on, like I mentioned before. When we're sitting in our chairs, we always have a little bit of muscle contraction happening. This slide just explains that a bit. Also, just a reminder that if you are about to have surgery in the operating room, the surgeon's going to want your muscles completely relaxed. Remember, we talked about the idea that the anesthesiologist can give you some rocuronium or other skeletal muscle blockers to ensure that you have complete flaccidity or flaccidness of your skeletal muscles. Also, sometimes if we have a patient in ICU that has a severe lung condition called ARDS, what can happen to them is we can get in a situation where we just can't supply them with enough oxygen to keep them alive. And what can happen in those cases is these drugs like rocuronium or other neuromuscular blockers can be given to make all the skeletal muscles become flaccid and have no tone at all. What this will do is it will minimize the amount of oxygen consumed by the body, as you can imagine, because now all the skeletal muscles are relaxed and it lessens the amount of oxygen consumption by the body. A whole new section. This section is pretty light actually compared to what we just finished up. This is talking about how our body moves. So we know that we have muscles that allow movement, and the muscles connect to our skeleton specifically in order to allow movement. So the origin is attached to the immovable or less movable bone, and the insertion is attached to the more movable bone, the bone that's going to be moved a lot. When the muscle contracts, the insertion moves towards the origin. So that what they're saying is that bone that the muscle is inserted on is going to move or contract towards the bone that has the origin of the muscle. Flexion and extension are opposite movements. Flexion decreases the angle of a joint and brings two bones closer together, as it says, and extension is the opposite. It's the straightening of, for example, your elbow or knees, and the term hyperextension is here as well. Lots of new terms in these slides. Abduction and adduction. The way that I remember the difference between these two is adduction moves the limb towards midline, that's true, and it's sort of adding together and getting closer to the body. So if that helps you remember. If you have a chance to twist your hand all the way in one direction and then twist it all the way in the other direction, you'll see that it will go almost 360 degrees. This is called circumduction, this movement of our hand in this circular motion. And actually, I find it kind of amazing that we have two parallel bones running down our forearm, our ulnar and our radius, and yet we are able to twist our hands almost full circle. These are just some of the muscles on the anterior of the body. We don't need to memorize all these, but here's a list of the ones that we should memorize. Masseter, trapezius, deltoid, tricep, bicep, quadriceps, and then on the patient's left, pectoralis minor, pectoralis major, intercostals, rectus abdominis, external and internal obliques, and transversus abdominis. Hey, okay, so these are some of the muscles from the posterior view. Again, we don't have to memorize all these, thank goodness. Let's focus on sternocleidomastoid, trapezius, deltoid, latissimus dorsi, that should be, gluteus medius and gluteus maximus. And let's also take a look at the tricep. Let's make sure we know the tricep. And that's about it for there.